Hi everyone, Mrs. Below here. I'm just continuing from where I left off in the last video. Again, this video is focusing on the kind of overview of the cell cycle. And remember, we're focusing on eukaryotic cells, which is what our types of cells are as humans. They're more complex than prokaryotic cells because we have a nucleus and a lot of other different types of organelles. So again, this is an overview of the cell cycle. It can be broken down into kind of like 3 big phases, which then are further broken down into subphases. So for example, interphase is where the cell spends most of its time, and that's when the cell is preparing to divide. And interphase is broken down into G1S and G2. Those are subphases. And then we have mitosis, which is the division of the division of the nucleus and the DNA, and that's broken down into PMAT, prophase, metaphase, anaphase, and telophase. And then the last part is cytokinesis, which is the division of the cytoplasm. And usually in terms of timing, that will overlap with telophase, which is part of mitosis. So let's focus on interphase first. So this is the time in between cell divisions, and it's when the cell is preparing to divide. So if you were to look at this cell under the microscope, it doesn't really look like anything's happening, even though there might be a lot going on. So interphase is where the cell spends most of its time. As you can see here, it is all of that time, which is a lot. And this can be further broken down into 3 sub-phases. So we have G1, which stands for gap 1. And this consists of growth, right? You're going to go from 1 to 2 cells. So the cell has to double in size. And all organelles increase as well, because if you're going to go from 1 to 2 cells, you literally have to double everything. So you're going to be making more mitochondria, smooth ER, rough ER, etc. And also the cell is just carrying out its normal everyday cell functions. Maybe as a skin cell, it's making keratin, things like that. A nerve cell, it's transmitting messages, etc. Well, that's a terrible example, a nerve cell. I'll get to that in a second when I talk about this part. But like a skin cell is, making keratin, things like that. The cell's just doing what it normally does. S phase stands for synthesis, and this is when the DNA replicates. Right? This is when you copy your DNA, which we talked about last semester in our DNA to protein unit. So all of your DNA goes through DNA replication. You unwind that double helix. Let me find the slide from last semester. So this right here, this is what's happening. You're unwinding that double helix. You're using helicase to do that. Use DNA polymerase to build a new complementary strand. And this is just a few bases here. I mean, we're talking millions of bases, and we're talking about all 46 of your chromosomes are being copied. And everything will be identical. And then the third phase of interphase is G2 or gap 2. And the cell is just continuing to grow and just preparing to divide. There is one phase that was not mentioned up here, and that is called G0 or gap 0. And this is when the cell stops dividing. This is when the cell literally will exit the cell cycle and it just stops going through this process altogether. Some examples of cells in the body that do this are nerve cells, brain cells, and heart muscle cells. And that makes a lot of sense. This is why brain injuries, spinal cord injuries to the central nervous system, and even injuries to the heart are so devastating. Your skin, when you get a cut, those skin's cells replace themselves, no problem. They're going through the cell cycle. But all of those nerve cells, brain cells, and your heart cells, like once they're damaged, they don't repair themselves, like in your central nervous system, and in your heart, and that can lead to like permanent damage. That's why brain injuries, spinal cord injuries, the central nervous system, and anything injuring the heart can be so devastating because those cells, they're in G0. They're not going to divide and replace themselves if they've been damaged. Not a fun thing to talk about. Also, we'll get into another not fun thing to talk about, but it's also just very interesting. So the cell cycle is controlled by a lot of proteins, okay? And one way to make sure everything is going smoothly and according to plan is we have these 3 main checkpoints, okay? And for example, in G1, there is a checkpoint where we're checking to make sure, does the cell have enough nutrients to grow? Is there enough growth factor? Do we have the things that we need in order to make the cell grow? Is the DNA damaged at all? Because we don't want to replicate a cell that has damaged DNA. That would be bad. So you can see this diagram has G0, which is when the cell exits the cell cycle. We have another checkpoint in G2. We make sure that the cell is big enough to divide, and that during this phase, DNA was replicated properly and we didn't make a mistake, right, and get a mutation. And then in mitosis, this is mitosis. This is the in the PMAT part of mitosis. This is the M part, metaphase, which we'll talk more about later. We're checking to make sure everything is lined up correctly and things have attached properly. Now, the thing is with cancer cells, the reason why cancer can be kind of challenging to deal with is cancer cells are cells that have been damaged, but they're not being stopped by these checkpoints. So you have a cell that's continuing to divide that's been damaged, and something's wrong with it. So you get this buildup of cells that are all damaged, which could lead to something like a tumor. So normally, so cancer occurs when damage has been done to genes. So the part of your DNA that's making those proteins that regulate cell division have been damaged. So the part of your DNA that is making proteins that are controlling this process have been damaged. So basically these checkpoints are kind of obsolete for that cell at that point. And normally if you have damage that's done to your DNA and it can't be fixed, or there's a mutation of some sort or whatever, normally what would happen is something called apoptosis. Which is programmed cell death or cell suicide. So essentially, a cell will self-destruct if it is beyond repair. Now, that's what a normal cell does if there's been damage done to it. Cancer cells, however, they have had damage done to the part of their DNA that's controlling the cell cycle. And because that part is affected, there is no mechanism in place. There are no checkpoints in place like stopping this. There's nothing stopping those cells from continuing to divide. And you actually have about 50 billion cells that go through apoptosis every day. So possible cells that could have become like mutations of some sort, something wrong with them, are actually being like gotten rid of on a daily basis. Also, normal cells stop dividing usually once they come in contact with each other. It's called contact inhibition. So like when cells are growing and they're all kind of touching another cell, they're like, okay, I can stop dividing. Cancer doesn't do that. So cancer, again, is controlled is when damage has been done to the genes that make proteins that control the cell cycle. And also, cancer can be characterized as uncontrolled cell division. Nothing is stopping these cells from dividing. So these cells will form a mass or growth, which can become like a tumor. And there's two different types, two general different types of categories we can put them into. One is benign. This is usually the term if you hear, obviously cancer sucks. And, if you've ever had someone go through this or been through this yourself, it's not a good thing, but this is just some really good, interesting information for you guys to know. So benign is usually the not scary kind of cancer. That means that the cancer is in a cluster and it's staying together and it can be removed with surgery. So that's usually the non-scary type. The one that's a little more scary is called malignant. And malignant is when these cancer cells tend to metastasize. And that means that those cells are not staying together or clustered anymore. And basically they can move around. And unfortunately, if they break off, they get into the bloodstream or the lymphatic system, which runs parallel to the bloodstream, it can end up in other areas of the body. And basically each type of cancer is just named for where it is, like breast cancer's in the breast, lung cancer's in the lungs, pancreatic cancer's in the pancreas, colon cancer's in the colon, so on and so forth. And that's why when some people get diagnosed with cancer, if it is malignant, it can spread and get in other places in the body, and that's usually the scarier situation. But essentially, cancer is uncontrolled cell growth, cell division, and the reason for that is because damage has been done to the part of the DNA in that cell that is controlling the cell cycle. So you can't control this anymore, so you get a bunch of cells growing that are damaged.